Got it.
You got it.
Woo.
Don't be staring at the door. Just go smooth.
Behave.
Alright, so this. Here we go. Here's naked people.
Naked babies right now.
Yeah, every 5 seconds.
Yeah.
I know.
Okay. So,
I'm Justin Bieber.
this picture shows how making sperm is different from making eggs, but it's hard to understand through this picture, so I'm going to show it right here. Now, if we go through meiosis, again, I'm going to erase the nucleus here. And I'm not going to draw as much stuff here because it kind of takes a while. I'm just going to move the chromosomes around for you. But remember, in prophase, they coil up so you can see them, and then they find their pair. Remember that? Their homologous pair. And the crossing over occurs, and all that jazz. And then in metaphase, they line up in the middle. Remember that?
Yep.
And then in anaphase, they move to opposite sides. And remember, they can assort themselves independently, independent assortment. But they end up on opposite sides. And then here's the big thing. In telophase, we end up, after cytokinesis, split down the middle. Right? But that doesn't happen in female humans. What happens in female humans is the split is uneven. Instead of splitting straight down the middle, it splits kind of like this. Where one cell gets most of the cytoplasm and the other cell is very small and reduced. We call this small and reduced cell the first polar body. And it will die. It will not survive. It's just eliminated. It doesn't have enough cytoplasm to sustain itself. There's not enough mitochondrions and such to produce the energy it needs for this cell to survive.
And that's called the polar body?
That's called the first polar body.
Does it ever live?
It doesn't live.
Why does it do that?
It's never lived? Is it just to get rid of the other 23?
No, what it's for is so that the surviving egg cell is big and gets plenty of nutrients.
Like, couldn't it just, like, send out the...
So right there, it selects certain genes and whatnot.
Are chromosomes too big just to, like, exit itself through, like, exocytosis?
Yes, they are pretty big.
So you can, so that's why that's...
It could get rid of them by exocytosis, but it doesn't. It just divides unevenly instead. Remember, these cells aren't thinking things. They've evolved to do it this way. So we go through a second set of division, which is called meiosis 2, in which these cells divide up. And this one's already dead, so let's not even worry about that. And this second one will divide up like this and divide its chromosomes. And then it'll split in two, but it also splits unevenly. And there's a pinching inward here and here. And that's called a second polar body.
Does it die?
And it dies too. So the female only has one surviving cell out of the 4. I mean, when one surviving cell out of the 3 here. But when a man makes sperm, he has 4 surviving cells that all become sperm cells. When a woman makes eggs, she only has 1 surviving cell. And it's real big. It gets all the cytoplasm. And that will sustain the cell as it goes through its first divisions, all this extra cytoplasm, because it divides really quickly. It doesn't have time to grow. Cool.
Is this sperm just like a messenger kind of?
Sperm just brings in
Is
the
that
chromosomes.
Yeah, so that's all it does? Or?
Yeah, it brings in the chromosomes.
Okay.
Yeah.
So is that 4 cells or is that just?
It's 4. I mean, usually this one dies before it gets to divide. If it does divide, there'd be 4.
I see.
Watch out for the table. I'm not answering that. They can leave a message.
I see. So if the first polar body divides, that'll be.
If
4
the first polar body there would divide it, it'd be 4.
I see.
There'd be 4 here.
Why do they keep
And
dividing?
sometimes that happens, but often it doesn't.
Why do they keep dividing?
Why do what keep dividing?
Polar body?
They don't.
They die.
They don't.
Wait, could the second
if
polar
they were
body
to
divide
divide,
before it
there
dies?
would've
Or only the first?
The second polar body?
Could it divide before it dies, or only the first one?
No. It doesn't have any chromosomes to divide.
Okay, never mind.
Yes, question?
So the sperm cells make 4 when they duplicate?
Yes.
And this makes 3.
No, this makes 1. These cells die.
Well, it would make 3, but
It would make 3 unless this one divided and continued to divide, and then you'd have 4.
Would that like cause diseases?
And sometimes that could happen, where the polar body lives, and then you have 4, but it usually doesn't happen. And so you can see up here, there's the first polar body,
If that does happen, do they eventually just die out?
and there's the second polar body. Yes, they would both die. There's not enough cytoplasm there to sustain them. The cytoplasm has all the mitochondrions needed to make the ATP, needed to let the cell do stuff. And if you have very little cytoplasm, you can't survive as a cell. This cell has a lot of cytoplasm though, and it survives really well. Yes?
are as many egg cells made as sperm cells?
No. Men make about 200 million sperm cells a day. About 2,300 a second. Women, all your egg cells are made actually when you're a fetus. And they're sitting in your ovaries right now. And there's thousands of them, but you'll only release about a few hundred of them in your life. 1 a month. Until you get about 50 years old, and then you'll
1
quit.
cell?
1 cell, 1
So
egg
we
cell
all
a month.
make them like right now?
No, you're not making them. They've all already been made.
Are people born without eggs?
No, they're all made when you were a fetus.
No, but what if there was fermentation?
Oh, yeah, I suppose that might could happen. Yeah,
People who can't have babies.
yeah.
Why? Or how?
There's lots of reasons why a person might not be able to have a baby. Maybe something's wrong with the ovaries. It can be cancerous or something. Maybe the little tubes that the eggs have to go through get blocked. There can be diseases and stuff that eat up the reproductive system. There's all sorts of reasons. Some guys sometimes have low sperm counts or sperm that don't form right. There's all sorts of things. So infertility, there's hundreds of reasons. You got to go to a fertility doctor and they try to figure out why. Sometimes they can give you drugs and make you release eggs if you're not naturally releasing. Sometimes they give you too much drugs and you release about 10 eggs and then you're like Octomom. 8 eggs. Okay? Does this make sense? It's just the way it works, the difference between men and women. So there's naked people right there. You want me to turn?
Whatever you're ready.
You know that's the first picture you looked at when you got the book. Here's a little video footage of sperm being made inside a man's testes.
Spermatogonia are the cells from which sperm cells arise. The spermatogonia divide by mitosis. One daughter cell remains a spermatogonium, and the other becomes a primary spermatocyte. The primary spermatocyte divides by meiosis to form secondary spermatocytes. secondary spermatocytes divide again to form spermatids. The spermatids differentiate into sperm cells.
Are they going back?
They have to back out of their little parking space.
That's cool.
2,300 per second for guys. And they continue to do that throughout their whole lives.
How?
100-year-old men are making sperm cells and can have babies and have before. The oldest father is like 103. Yeah.
How big is like one of these cells?
Tiny. You need a microscope to see a sperm cell. The egg cell, though, is the largest cell in the human body. And it's the size of a period at the end of a sentence in your book.
So,
The human egg cell. It's the size of a period at the end of a sentence in your book. That's how big a human egg cell is.
Like a woman egg cell?
A female's human egg cell. Guys don't have it. That interesting? Okay, let's talk about some problems that can happen. Some mutations that can happen and lead to problems. One mutation is called a non-disjunction. Let me turn the light on for this so we can see it pretty well. I need a remote control light switch.
I was supposed to have one of those ones where you like clap your hands.
Clapper.
The clapper.
Clap on.
Clap on. Clap on. Clap on. Clap on. The clapper. When did you get the clapper? Shore Leave. What movie? Austin Powers.
Which Austin Powers was that?
The first one, I would think. Yeah.
One day.
One day.
That day.
Alright. Let's see some of the things can happen. There's a mistake that sometimes happens in cells. It's called non-disjunction. Let me show you a non-disjunction. Here we have the chromosomes during prophase. They pair up with their homologous pair. And then they get in the middle. Everything's going good so far. You ready? Anaphase 1, they go to opposite sides. Telophase, of course we divide. I'm not going to draw all the nuclei, it takes too long. And then the second division, here we go. They line up. They all line up. Everything seems to be going well so far. They separate during anaphase. Great. Separate. Separate. Mutation. This one never separated right.
So what happens?
Wait! So this will form into a cell here, and so will these. And they'll become sperm cells. They'll grow their little tails. And if this sperm fertilizes the egg, that's great. It's got the right number of chromosomes. And if this sperm fertilizes the egg, that's great. It's got the right number of chromosomes. But if this sperm fertilizes the egg, it's got one chromosome too few. And if this sperm fertilizes the egg, there's one chromosome too many.
Down syndrome.
A Down syndrome child has one extra copy of chromosome number 21. And so we call this mistake a non-disjunction. And it usually leads to death of the organism. Almost always when you're missing a chromosome, the organism will die. And almost always when you get an extra chromosome, the organism will die. It won't. The first cell won't start dividing. It will never form into a human being. But if it's an extra number 21, the organism lives, grows into a human being that has Down syndrome. We call this error a non-disjunction. There's the word right there. A non-disjunction is when the division doesn't take place evenly. And it can happen in two places. It can happen in meiosis 1 during anaphase when they're separating, or it can happen in meiosis 2 during anaphase when they're separating. Either way, you can end up with a non-disjunction and have extra copies.
So,
Yes?
how common are those mistakes? Because it would seem like if they happen like somewhat with like any like visible frequency, that would have to, like something like that would have to happen a lot, does it?
It's pretty uncommon, but it's not as uncommon as you'd think.
Okay, is it like, there's one of those I have like once a day or something, or like once a month?
Well, if a guy's making 2,300 sperm per second, and it happens in 0.1% of the sperm, that means there's about 20 mistakes per second like this.
Okay.
That's a lot of mistakes. 20, 20, 20, 20, 20. I'm counting the number of non-disjunctions going on in my cells. It's a lot. Now, luckily, there's so many that are made correctly that the odds are that one of them won't fertilize the egg. And there's plenty of times where a screwed up chromosome does a screwed up sperm does fertilize the egg, and then that thing just dies there, and the woman never gets pregnant. And she just goes along with her life and doesn't know what happened. And the couple says, well, we've been trying to have a baby. You know what that means, don't you? They've been having sex every night. Next time someone says that to you, we've been trying to have a baby for 4 months. Say, you've been having sex every night for 4 months? See what they say. So, usually the thing dies, but in some chromosome combinations you end up with Down syndrome. Or some other mistake. And here's a karyotype that you see here up at the top. You can see the chromosomes have been laid out by a computer. And yes, they have 2 copies of number 1, 2 copies of number 2, 2 copies of number 3, and so on until you get to 21. 3 copies of 21. That's one too many. And we have a name for that. Whenever you have 3 copies of a chromosome, that's called, let me erase this, right? 3 copies of a chromosome is called a trisomy. And that causes Down syndrome, which there's a lot of problems with the Down syndrome child. They have mental retardation. They have a cleft palate. They have different their joints or flexibility of their joints are different. It affects the whole body. They don't live as long. What's wrong with this girl? She's got Turner syndrome. She looks somewhat normal.
She's underdeveloped. Well, she'll never go through.
She's missing an X chromosome. So what happened was the X chromosome, it's like this sperm cell, if the X chromosome went down here, then this sperm cell's missing an X chromosome. And by the way, the X chromosomes determine your sex. If you have 2 X chromosomes, you're female. And if you have an X chromosome and what's called a Y chromosome, which is much smaller, you're male. So, she's just got 1 X. And that means she's female, but she's got Turner syndrome. Turner syndrome ends up making a female with a problem. And what problem do you have when you have Turner syndrome?
Right here in the book.
It says in the book. Let's take a look.
A short, broad chest and widely spaced nipples.
Turner females are short, broad chest, widely spaced nipples. These individuals have a low posterior hairline and neck webbing. The ovaries, oviducts, and uterus are very small and underdeveloped. Turner females do not undergo puberty or menstruate, and their breasts do not develop. However, some have given birth following in vitro fertilization using donor eggs. They usually are of normal intelligence and can lead fairly normal lives if they receive hormone supplements. There you go.
They don't go through puberty.
What's that?
They don't go through puberty.
They don't go through puberty. 1 in 10,000 females ends up with Turner syndrome. I know there's some people who realized that they had these syndromes once they took the biology class, and not until then.
Are you serious?
Uh-huh.
Wait, what?
I've heard about that. I've never taught one, but I've heard about it. I've heard about kids are learning about this, and they go, "Wait a second, that looks kind of like me. I have those symptoms."
It's really awkward.
And they never realized that they had
Wait, they don't
this
know like
syndrome.
at birth that they have it?
Not necessarily.
But what if they like go to the doctor and the doctor would explain to them?
Not necessarily. Some people don't go to doctors.
It's really weird.
Some people are born and they live their life and end up at school somewhere and figure this out. Klinefelter syndrome, that's a XXY. So they got an extra X. That's a trisomy. Oh, by the way, if you only have one chromosome, it's not called a trisomy. You know what it's called? Monosomy.
Can you have only one Y chromosome?
If you only have one Y chromosome, you don't the cell doesn't survive.
Okay.
It has to have an X to survive. So you won't see any individual with just a Y. But if you have 2 X's and a Y, you will survive and you'll grow up to have Klinefelter syndrome. A male with Klinefelter syndrome, okay, in Klinefelter males, the testes and prostate gland are underdeveloped and facial hair is lacking. They may exhibit some breast development. Affected individuals have large hands and feet and very long arms and legs. They are usually slow to learn but not mentally retarded unless they inherit more than 2 X chromosomes. No matter how many X chromosomes there are, an individual with a Y chromosome will be male. While males with Klinefelter syndrome exhibit no other major health abnormalities, they have an increased risk of some disorders, including breast cancer, osteoporosis, and lupus, which disproportionately affect females. Although men with Klinefelter syndrome typically do not need medical treatment, some have found that testosterone therapy may help increase muscle strength, sex drive, concentration ability. Testosterone treatment, however, does not reverse the sterility associated with Klinefelter syndrome due to the incomplete testicle development. There's a whole section on top of page 186 that talks about Klinefelter syndrome.
So it's just like girls and guys with sex features?
It's a guy that never developed, his male features didn't develop.
So what was lupus they mentioned? Is that something?
What's that?
Lupus? What's that? I never heard.
Lupus. I don't know what lupus is.
Okay.
Here are all of the symptoms of Turner syndrome.
They get measles?
Brown spots, nevi. They're not measles. They're just brown spots on the skin. And here are the symptoms of Klinefelter syndrome. This is a guy. He's kind of shaped like a girl. That's a guy. Guy. It's a drawing.
That's really
What's that?
Would they like freak what? This is
Would they what?
how they like? Is this like? They like turning like hermaphrodite?
No,
Not
hermaphrodite, you have
necessarily.
both.
Yeah. How do you get hermaphrodite?
I don't know about hermaphrodites. Okay.
That would be a guy.
What's that?
It would be a guy.
It'd be a guy because he's got a penis.
Hermaphrodite has both.
Yeah.
Both sexes.
So would a hermaphrodite be like an it?
I don't know what a hermaphrodite is. Look up hermaphrodite on Google tonight. I'm sure you'll get a lot of interesting.
Just normal people, like a normal guy with like an ovary inside of him. Doesn't make him any different. Or you could
No,
be
a woman or girl for
has
the,
it.
yeah.
Yeah, that's what I'm saying. It could be either. But it's not like you're like manly. You just have an extra organ inside of you.
Well,
Not
some,
one. That one rather one.
Okay.
Sometimes they're different.
I've got some other chromosome mutations to show you. Get these magnets out. And your book shows them up here, and so I have little magnets that kind of demonstrate. Let me put this together just a second. Sometimes the chromosomes okay.
How did she fit that ring on?
Now, so what I've done here, turn the light back on for this. What I've done here is we have our chromosomes here. And I've simply taken some of them here's our cell. Guess I could have done this on the other side. Here's our chromosomes in our cell, and I'm just going to take these white ones out and put these purple ones in their place, these orange ones in their place. Okay? And I'm going to take one of these green ones out and put this green one in its place. Okay, so I've removed one of the chromosomes just to demonstrate some of these other mutations. Is that okay with you guys?
Yeah.
You would be highly upset if I did that. You would have no choice but to sit there and enjoy. A deletion. A deletion is simply this. During the movement of these chromosomes around, sometimes you can get a piece, whoop, falls off, and is lost. Floats around somewhere and maybe ends up getting stuck in the membrane or something, and it's just not used anymore. Maybe it gets destroyed by some lysosomes. That's called a deletion. You see up there, A is a deletion. Right here. The little piece came off the top. That's why they're showing that A there. It broke off. That little A broke off. And so the arrow made you go from here to here. Do y'all see?
Yeah.
That's a deletion. What about a duplication? A duplication is when you take a piece. Watch the chromosome here. Remember, these are exact copies. Remember the ones that are stuck together are exact copies of one another? Y'all remember that? This piece and that piece are exact copies? Well, what if one of the exact copy pieces slides over like this onto that one? Then we're repeating information on the chromosome. This chromosome is a large area of instructions for the cell, and we've repeated it. That's possibly not a good thing. It'd be like reading along in your book and you have chapter 12 and then chapter 12 again. If it was you, you just skip it. You go, oh, they just repeated chapter 12. But the cell's not smart like that. The cell will read it again. It might make extra stuff that's not necessary.
Is it always bad?
Almost always. Sometimes a mutation can be good. But in a well-adapted creature, it's usually bad. But if the conditions change, a mutation might be favored. We're going to learn about that. That's evolution. But for most part, it ends up being bad. So that's a duplication. And that's shown right here. The DE part is duplicated. Another DE.
Does it get duplicated, like taken from one side?
Yeah. One of the two sister chromatids, one of the pieces comes from it and switches over. You see.
Okay.
So the other one has a deletion.
Okay.
So this one has a deletion, and that has a duplication.
Okay.
Nice. What else? Inversion. Check this out. What if this happened? Boop. 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 That's an inversion. Think that would screw anything up?
Yeah.
You think if you were reading along in your book and one of the chapters was put in upside down and backwards, that would mess you up a little bit?
Yeah.
Especially if you were stupid and just kept on reading?
Yeah.
Cell's kind of stupid. It doesn't have a brain where it's thinking. It just reads the instructions, whatever's there. So that's an inversion. And finally, a translocation. This is a real interesting one. Here's a translocation. When a piece comes off of one chromosome and a piece comes off of another non-sister chromatid and switches that'd be like you're reading along in your book, in your biology book, and then there's some history pages in there. You're reading along in biology, and there's physics. You're like, whoa, this makes no sense, because it's from a completely other chromosome that has completely different information. That'd screw things up too, don't you think? Do you want to look at some of the screw-ups?
Yeah.
Probably not politically correct. All these people screw up.
Wait, translocation? What's it do?
Translocation, watch. Who's asking that? Here's the normal chromosome. Translocation.
But can't, like, the white and green from there? Oh, no, like, those came from, is that if they came from, like, the same source? Because the white and green chromosomes can switch pieces.
Is that like just crossing over?
Yeah, that's true. If this was imagine this was a smaller chromosome, a non-homologous chromosome.
Okay.
Yeah, good thinking. Yeah, if these were homologous pairs, it'd be just like crossing over. But let's say they're not homologous. Say it came from the little one. These aren't homologous pairs, so if a piece switches like that, you got a big mess. You got a big mistake. Good thinking, Ella thinking. I like that. Here, let's watch a quick video of this kind of thing happening, and then we'll take a look at people with the disorders.
A chromosomal mutation is a substantial change in the structure of a chromosome or chromosomes. One way that this can occur is by a deletion or a duplication within one chromosome. In the chromosome, shown here, the longer arm is labeled Q and the shorter arm is labeled P. Regions are designated with numbers. If a region is deleted, such as a large portion of the Q2 region shown here, then a deficiency occurs. There is now less genetic material in the mutated chromosome. A duplication occurs when an extra copy of a chromosomal region exists, such as the extra copy of regions Q2 and Q3 shown here. Both duplications and deletions can be created by abnormal crossing over during meiosis. An inversion occurs when a chromosome is broken in two places and reconnected so that a region is flipped from the normal order, such as the Q2 and Q3 regions shown here. Note that an inversion does not change the total amount of genetic material in the chromosome. Translocations involve two chromosomes. In a simple translocation, a single piece of one chromosome breaks off and is attached to another chromosome. In a reciprocal translocation, two chromosomes exchange pieces. Note that translocations do not change the total amount of genetic material. However, both inversions and translocations can lead to overall deficiencies and duplications of genetic material after meiosis.
Okay?
What causes those things to switch?
I don't know. You know, when you're shuffling pieces around, doesn't always work. You ever been shuffling cards and one flies out? You know, you got a lot of moving parts here.
Just happens.
And so it'll happen. And so, you know, during crossing over, during normal crossing over, pieces are breaking off and switching. So maybe if some of that becomes loose or something, it could float around and end up somewhere else. That's a good question, though. I don't really know the answer. Not sure why that happens, but it does. So these girls have Williams syndrome. Williams syndrome is a deletion of a little piece of a chromosome. The very end of a chromosome falls off and is lost. And they even know what chromosome it's on. It's the end of chromosome 7. So let's go back to our, here's our karyotype. There's chromosome 7. So if you're missing a little end piece of chromosome 7, you'll have Williams syndrome. You don't get to choose, you'll have it. And what are Williams syndrome kids like? Well, children who have this syndrome look like pixies, with turned up noses, wide mouths, a small chin, and large ears. Although their academic skills are poor, they exhibit excellent verbal and musical abilities. The gene that governs the production of the protein elastin is missing, and this affects the health of the cardiovascular system and causes their skin to age prematurely. Such individuals are very friendly but need an ordered life, perhaps because of the loss of a gene for a protein that is normally active in the brain. So they're missing some genes, they're missing some information, and that has strange effects. They seem happy, except for him. Williams syndrome.
So they aren't smart, but they're like musically inclined.
Yeah. Maybe some of the great musicians in history had Williams syndrome. Who knows? Could be. Translocation syndromes. This is the one y'all were looking at yesterday. Figure 1014 shows a person who has a translocation syndrome between chromosomes 2 and 20. Although they have the normal amount of genetic material, they have the distinctive face, abnormal eyes, abnormalities of the eyes and internal organs, and severe itching characteristics of Alagille syndrome. People with this syndrome have a deletion on chromosome 20, which can lead to a congenital heart condition called tetralogy of Fallot that produces digital clubbing of the fingers. That's what this says, I guess.
Hi everyone, just two quick reminders. If you are going on the college trip tomorrow to Jacksonville, please make sure that you bring Ms. Nevins your mission slip today. And also, don't forget that High Point University will be in the Schumann Conference Room at lunch today. Thank you.
So they have a translocation between a couple of chromosomes. Let's see if this shows anything.
In all the copying of the thousands and thousands of base pairs of nucleotides during cell division, it is possible for errors to occur. When these types of mutations occur, serious problems can result. Cystic fibrosis occurs when a certain part of the gene is incorrect and the protein misfolds. Down syndrome is the result of an extra copy of chromosome 21, resulting in a total of 47 chromosomes rather than 46. Sickle cell anemia is a genetic disorder brought about by a one-letter misspelling of a base pair in a gene that results in defective red blood cells. Cells have ways of correcting errors. A type of proofreading function scans the DNA and corrects any base pairing mismatches by removing the improper nucleotide and replacing it with a proper one. Some mutations occur spontaneously, but others can occur as a result of exposure to physical or chemical agents. Cancer is a mutation of the cell mechanism that regulates cell growth. It can be caused by such things as cigarette smoke, ultraviolet rays from the sun, exposure to asbestos, medical x-rays, and other natural and environmental causes. Once a cell has become cancerous, it can divide, repeating the cancer in each new cell that is created, which spreads the disease. But not all mutations are destructive. Over the course of evolutionary time, mutations in the cells of organisms have led to genetic diversity and the rise of new and more competitive species, better adapted to their environment.
So mutation is an important part of evolution, which we're going to learn about. And here are the other sources of variation that we studied. Crossing over, independent assortment, and sexual reproduction. The purpose of sex is for variation. We talked about that when we were doing meiosis. You get more variation of the offspring. So, one thing that you might want to think about is, gosh, I'm pretty lucky to be alive and have the right number of chromosomes. You're just one chromosomal error off from not even being here or having something like Down syndrome. And so it's the laws of chance that govern your life. Sometimes people end up on the wrong side of that. Yes?
None of these are like hereditary.
They would only be hereditary if you could still have kids with the disorder. Now, Down syndrome people can't have kids, and a few of the other ones we looked at can't have kids. But if you could have kids, you could pass on to them the chromosome that has the problem and give it to your kid. Yeah, it's possible.